Go ahead, Joe, you are live.
Okay, thank you. Good evening. Welcome to the November 2021 meeting of the Oregon Mycological Society. I'm Joe Cohen, the current president of the society. This has, as all of you know, been a very challenging year for us because of a combination of COVID and continued very bad drought conditions. We've got through the year and hopefully we will look forward to better things next year. I do, however, want to give a special shout out to two people, Kim Brown, who's kept our monthly meetings going with an excellent cast of speakers, and to Kim Robinson, our field trip leader coordinator, who has kept our field trips going, even though she has been out of state most of the fall. And when I looked at the numbers of field trips we had, I was actually very pleasantly surprised. It would not shock me to learn that we had more field trips this fall than in any other fall. So I hope we will continue to see all of you at monthly meetings and as we hopefully resume live events at those events. And with that, I want to turn things over to Kim Brown, who has something to say about elections, and will then introduce our speaker. Thank you, Kim.
Thanks, Joe. So hi, everybody. Hope that the fall mushrooming has treated you well. I mean, initially, that's why we joined the club and that's why we're all still here. We're all fungophiles. So with that, I want to say that the OMS elections are currently ongoing, and that's for the officers and board of directors for next year. First, I want to thank two people. I want to thank Jenny Housechild and Maggie Iadanza for being on the nomination committee and supporting these elections. We have switched, obviously, for the second year in a row from the live elections to the digital ones. The instructions were sent out to the email that we have on file for you on the 3rd of November, and the voting opened then. And it's open through until Wednesday, the 10th of November at 11:45 p.m. So current members, as I said, you should have received your electronic ballot to the email address that you actually provided. Family memberships will receive 2, so long as there's 2 email addresses attached to those that were provided when you registered. So if you don't see the ballot in your inbox, go back and check, sent out on the 3rd of November. Check your spam folder, junk folder, or any other email filters that you have. And just note that it was not sent by MailChimp, but actually from Election Buddy. So look for that on behalf of the Oregon Mycological Society. So it'll say invitations at mail.electionbuddy.com. So if you didn't receive a ballot and you are a current member, please go ahead and check your membership again. Or if your family membership only received 1 instead of 2, actually go ahead and please email membership@wildmushrooms.org. That's membership@wildmushrooms.org. And we have a great slate sent up. I am going to just go ahead and share my screen. Let's see. And pull that one up. And let's see. Joe, can you see the candidates, the list of candidates there?
I can see it in Zoom, but not yet in, hold on, it's about, there it is.
There
yes,
it is.
I can.
So good. So this is the lovely smiling faces of everyone who is on the ballot this year. Your choices, we have a number of incumbents you'll see for Joe for president, Shayna, Dawn, Kim Robinson is an incumbent. But you have a number of choices. 2 great people who have stepped up for vice president, Corey Lucas and Shayna Hurst, and 3 people who have stepped up for the 2 director positions, Kim Robinson, Ethan Disbrow, and John Thompson. Great people. My apologies to John. I just realized that I had a typo in your name, but you'll see that the name corrected in the ballot. So there again, those are our candidates. So please go ahead and vote. And I look forward to seeing what we come up with. Going to be a great year next year. Hopefully lots of things that we can bring to you guys. So I'm stopped sharing my screen now. And I am going to introduce our speaker. So we are honored tonight to present Dylan. It's Baldassari, right, Dylan? Nod your head. Yep. Okay. One of our 2020 OMS scholarship recipients. And we're very proud to be able to support him and his research. Dylan's passion for the outdoors and fungi sprung from growing up and spending large portions of time in the outdoors in southern Wisconsin, and learning vociferously about the natural world through books, magazines, and mentors. The passion for the outdoors and the environment then led him to pursue a bachelor's in biology with minors in chemistry and statistics. Ooh, statistics, there's a hard one for you. At University of Wisconsin in La Crosse. While working on his bachelor's, he took Tom Volk's mycology and medical mycology courses. And from anyone who's familiar with Tom's teaching style or have seen him when he presented here a while back, you would definitely understand from that on how he got hooked on fungi. Dylan eventually ended up working on his master's degree at the university with Tom Volk and Todd Osmundson's guidance. Tonight, he'll be sharing the findings to date of his research, attempting to unravel the relationship between fungal endophytes, whitebark pine, and the invasive fungal pathogen, whitepine blister rust, that is decimating the whitebark populations. And there you go, Dylan. Really quick to everybody who is on YouTube, go ahead and we're going to be having the questions that are going to be answered by Dylan at the end of his talk. So please go ahead and write your questions in the chat and I will be moderating. With that, thanks, Dylan. Take it away.
Thank you, Kim. That was an awesome introduction. And thank you to the Oregon Mycological Society for supporting my work. I really, really appreciate it. And I'm so excited to share what I have today. So let me pull up my presentation and we'll get to it. All right, so my screen should be sharing, should be good to go. So I am presenting on fungi in the sky, fungal invaders, defenders, and reserves. So I think I should start by, all right, let's make sure I can, there we go. All right, I think I should start by describing this somewhat cryptic title a little bit. So why fungi in the sky? Well, this sampling took place at around 9,000 to 11,000 feet. So these are high elevation fungi that we're looking at. Also, all my sampling took place in Montana, so it's big sky country. And another sort of link to it is that one of my sort of catalysts for wanting to study this or for thinking about these kind of systems, these high elevation systems, was having a background on the Sky Islands in Arizona, where they look at population genetics using these island mountain ranges. And that's something that we're going to get into later. And what are fungal invaders, or who are they in this case? Well, it's whitepine blister rust. It's this invasive fungal pathogen, which we're going to dive into in a little bit. And the defenders are endophytes, or at least potentially the defenders. In many systems, fungal endophytes have been shown to have profound impacts on the immune system of plants. And that's one thing that my study is looking at. And who are the reserves? Well, this is kind of my opportunity and my play at looking at fungal conservation. And specifically, are there other species of trees that can provide a reservoir of fungal symbionts for whitebark pine if whitebark pine populations continue to decline? Also, I would like to say thank you to the University of Wisconsin-La Crosse, obviously for allowing me to do a master's here, to Tom Volk, to Todd Osmundson. They've been tremendous mentors and have only stoked my passion for fungi. So fungi in the sky. Right here, we have an image of whitebark pine. This is in its Krummholz form. So this form is distinguished by its stunted growth. Maybe you've seen them like this. They're windswept, they're small, they're almost shrub-like, and they're very different from, the kind of iconic 1,000-year-old whitebark pines that you might be used to seeing. But they do play an important role, and it offers a really interesting system to study whitebark pine and to study the fungal inhabitants. So they are restricted to the subalpine, which is not unique per se for conifers. There are other species that are restricted to the subalpine, but whitebark pine has a number of other really important reasons to study it. So that's why I selected whitebark pine. And it also allows us to look at tree line advance, because whitebark pine is growing at the highest elevation suitable for any conifer. They are actually pioneering the tree line creep or the tree line advance. So we can actually see climate change happening in real time by pine trees establishing further and further up the mountainside. Real quick, an outline of what I'll be talking about today. I'm going to start by doing a background on the study system. We'll get into objectives, my methods, both in the field and in the lab, and then some of my field results that I have right now and my expected molecular results. Unfortunately, I've had some COVID-induced delays just being able to get the materials that I've needed to finish this molecular work. So I'm a little bit behind, but this last week I've been preparing my samples for Illumina sequencing, and they should be sent out soon. So I'm really excited to see what comes of that. And then we'll just kind of wrap it up with an overview and an outlook. So let's kick it off with the study system. We should probably start by talking about endophytes. I know that people's exposure to endophytes and fungal endophytes can vary kind of dramatically. So we'll talk about what are endophytes. And if you could do me a favor and just take a look at this image and just for a few seconds, think about what you see. So please, for a few seconds, just think about what you see in this image. All right, that's a few seconds. So for some of you, you might see a variety of conifers, right? You might see a whitebark pine, you might see, this spruce and subalpine fir forest below. If you're a skier, you might see some avalanche shoots, you might see some sweet drops that you'd like to take. Most of you are probably like, oh, that's a chanterelle spot that I'd like to go take a look at. I see this as, and I see any lecture or talk as an opportunity to provide, even momentarily, a new lens to look at things, and the lens that I look at things through. So every plant studied to date has been found to have fungal endophytes. These are endophytes that live inside the needles of pine trees. They live inside the leaves of all plants, and every blade of grass, every leaf, every pine needle that you see, they have fungi living inside of them. They are all over the place, and they are all connected and interacting with each other. I think one of the amazing things about fungi is, yes, they're so small, microscopic, and there's the humongous fungus out there, but most of them are really just operating at this microscopic layer, but their power is in their numbers. They're operating at a completely different scale than mammals or than humans. So again, what are endophytes? Well, they're ubiquitous. They are found everywhere in every habitat type on every plant, and they can be fungal and bacterial. In some cases, there's really unique and specific interactions that actually happen between the fungi and the bacteria. From what I've gleaned from the literature thus far, fungi appear to be playing the larger role here than bacteria. Though bacterial endophytes are important, fungal endophytes generally are having a larger impact than bacterial endophytes. They were described as hyperdiverse in a paper by Betsy Arnold and colleagues. And, it's kind of an interesting way to think about it. It's like hyperdiverse in comparison to what? Hyperdiverse in comparison to insects, Probably not. Hyperdiverse in comparison to even beetles? Probably not. Hyperdiverse in comparison to mammals? Absolutely. But in talking to her, what it really was about and why she described them that way is because it was this unprecedented diversity. Because they are cryptic and microscopic, they can't be seen with the naked eye. It really took the molecular revolution to allow us to examine endophytes. They're also asymptomatic. So if it's truly an endophyte, there's no way you can even see it visually just by looking at the leaf, right? You got to pull out your microscope, or you got to grow it on a petri dish, like you can see in these two photos above. And if it's not cryptic, well, then it's probably just a fungal pathogen, right? If it's, producing some signs or symptoms of actually being there. In this image on the right, you can see a petri dish with labeled cultures that are coming out, right? So they're numbered. And what's really cool about this is one needle that's been divided into 3 sections, and there's 8 different species growing out of that single needle. So there's 8 different species of fungi that were living inside of that needle. And what's even more interesting is that 7 of these species that are growing out of there are from the same genus. So their morphologies can vary greatly, right? And I believe that's the genus Lophodermium, which you'll hear come up many times in the world of endophytes. So when thinking about the plant microbiome, I often draw the parallels to the human microbiome, right? We have our gut microbiome, which I think is very analogous to the soil microbiome of plants, right? Our gut microbiome helps increase the bioavailability of nutrients. It produces acids and bases. It helps control our gut chemistry. It produces enzymes that help us break down food. It does so many important things. The root microbiome tends to do the same things, right? We're all familiar with mycorrhiza, the bacteria and fungi living in the soil that are freeing up nutrients, and in some cases, sort of acting as a conductor to get it to the plant, and in exchange getting sugars. Fungal endophytes or foliar endophytes are a bit more like our microflora, right? So we have these different patches of microbiome around us. Like we have a distinct oral microbiome, we have a distinct nasal microbiome, we have a distinct armpit microbiome. Well, they have a distinct leaf microbiome that does important things for it. In some cases, they produce toxins that are, they produce compounds that are toxic to herbivores. I think one of the most extreme examples that you've probably heard of is ergot or ergo poisoning and Claviceps purpurea, right? This sclerotia that's produced, and if you have a cow that eats it, the cow will probably die from gangrene and get a little acid trip along the way. That's an extreme example. In most cases, these fungal endophytes are producing low levels of a toxin that if a herbivore overeats it, it will get sick, right? So it prevents it from overeating its host plant species. They can also play a really interesting role in nutrient acquisition. You're probably wondering, how can that happen if it's just inside the leaf, right? It doesn't have any extraneous source to pull from. Well, it still has the atmosphere, right? So this is where some bacterial endophytes are really cool in that they can live inside of those leaves and they can be fixing nitrogen while they're in there. They can also improve drought tolerance, which obviously is super important with this changing climate that we have right now and is starting to be applied in agricultural systems. And they can improve disease resistance. And we'll get into that a little bit deeper. There's 2 primary ways that they can improve disease resistance. The first is by an immune response activation. So what happens is that fungal endophyte, it gets inside the needle, it gets inside the leaf, and the plant recognizes that something foreign is in there. And it recognizes that it's not really doing anything right now. It's not trying to take over or attack the plant, but it starts to prime its immune system. It starts to prepare, in the case of pines, the terpenes and the polyphenols that it might need if that fungus or that foreign entity ever decides to get out of control. The second way that they can prevent diseases from moving into a plant is by directly inhibiting pathogens, right? So that leaf or that needle is a niche. It's that fungus's home. So if you have another fungus that tries to move in there, it's kind of going to say, hey, this is my home. Like, this is my niche. You should stay out of here, right? So it can do that either through mycophagy, so just by consuming the fungus, or it can, sort of attack it with an enzymatic attack. If you look at this image over here on the right, which comes from Arnold and colleagues in a 2003 paper, you can see how fungal abundance is associated with this alternaria leaf spot on apple. So basically, the higher fungal abundance in specific species of fungi were correlated with decreased alternaria spot. So needle endophytes and how they mitigate disease. Well, Ganley and colleagues in 2008, they found that certain slurries that they inoculated on the needles of Western whitepine mitigated the severity of whitepine blister rust. In 2018, Lorinda Bullington and colleagues, they planted whitebark pine seedlings in a common garden, and they analyzed their endophyte communities twice. They analyzed them before blister rust inoculation and after blister rust inoculation. And what they found were that there were links between disease severity and endophyte community. To throw a bone to the endophyte enthusiasts out there and to the taxonomists out there, it was Lophodermium, species of Paraphoma, and Metarhizium and Nicefolii that were specifically correlated with decreased needle spotting and cankers. They also found that there were community shifts. This was likely due to the tree's immune response. When it recognized that it was starting to be attacked, it released a suite of chemicals inside of the needles to try and kill off whatever was there. Unfortunately, by that time, whitepine blister rust has already gotten into the vascular system. So killing off what's in the needles isn't actually going to do it any good. So whitebark pine in the system that I'm working in is really a cool one. As I said before, it's restricted to the subalpine. Here in this habitat distribution map, you can see how whitebark pine is just isolated to these high elevation patches of mountain ranges. And it has this patchy island-like distribution that ranges in habitat size and distance from one another. It's also a keystone species. So what does that mean? A keystone species has a disproportionate effect on biodiversity. So in particular, whitebark pine produces this calorically dense this calorically dense cone that has tons of seeds in it. It's a key food source for grizzly bears, for mesomammals, for birds. It's also a foundational species. So what does that mean? Well, whitebark pine really, is excellent at establishing in these high elevation environments. So it can establish on rocky soil where there's basically no soil, where there's extreme environmental conditions. It's very harsh. And when it establishes there, it starts to solidify the soil, preventing erosion, and it provides a microclimate. So other plants and other flora can move in there and start to establish. So just the establishment of a single whitebark pine or a few whitebark pines can eventually lead to an area that was previously just, rocks and shale being a beautiful forest and a biodiversity hotspot. It's also threatened. I haven't checked on this in a few months yet, but I know it was up for public discussion to be an actual ESA protected species. So we'll talk about why its populations are decreasing and what that could mean for ecosystems in the Mountain West. Whitebark pine as a keystone species, right? I said it was a key food source for grizzly bear. Take a look at this. this cool moving image of a grizzly bear. And what it's doing is it's digging up caches of whitebark pine nuts. So when Rocky Mountain grizzly bears wake up, they actually don't go into hibernation. They go into something called diapause, which is a lower metabolic state that lasts for 2 to 3 weeks. And then they wake up and they get out of their dens and they go and they look for carcasses and for caches of whitebark pine nuts. And I can't remember the exact numbers, but there's been some studies done on how many calories are, where their main winter calories are coming from. And whitebark pine nuts account for a huge proportion of that. This bird on the left, we're going to talk about right now. This is called the Clark's Nutcracker. It's really a beautiful and intelligent bird. It's a corvid, so it's related to crows. And I'm sure you've had some, reference to how intelligent crows are. Clark's nutcrackers are also super intelligent. And they're this beautiful example of evolution and how two species can be tied together. One other thing I just want to say on grizzly bears that popped into my mind is that a key speaker at a Whitebark Pine conference once said that the fate of the grizzly bear in the lower 48 is tied to the fate of the whitebark pine. It's that important of a winter food source for them. So the Clark's Nutcracker, to get back to these beautiful birds, are really the planters of whitebark pine. They're the gardeners of whitebark pine, and they're doing so in a variety of ways that just happened. Well, I shouldn't say just happened, evolutionarily are great for the whitebark pine. They plant these caches, right? Their plan is to go dig them up eventually, but they forget about them. And they plant genetically diverse caches. So they'll actually go around to different whitebark pines. They'll fill up their, I can't remember the name, I'm not a bird person. They'll fill up their throat with whitebark pine seeds from a variety of trees. So when they plant that cache, it's this genetically diverse cache. And some of those seeds might be more adapted to that particular environment that it's planted in, and therefore have more likely chance of survival. They'll also fly quite a ways to plant these caches, and they tend to plant them in last year's burn sites. So what an advantage for the whitebark pine, right? Like, you have this bird that's planting you in these burn sites that are wide open and ready to be taken over. So a very cool example of evolution. Why is the whitebark pine declining, and how much is it declining? Well, paper by Mohat et al. In 2008, in her sites, she found 40 to 90% population declines. The leading factors for this population decline, one is white pine blister rust. That's the primary antagonist. It's this invasive pathogen that this invasive fungal pathogen that evolved in Asia. Eventually it moved over to Europe. It had, pretty severe effects on the stone pine population in Europe. And then eventually it got moved to North America as well. Climate change is also a key factor in this. It comes as no surprise. There's a variety of reasons why climate change is affecting whitebark pine and affecting these ecosystems. But one cool one that I like to think about is this shrinking habitat, right? So as, if you think about a mountain, it's this, it's essentially like a vector, right? And at the bottom of the vector, you have the most amount of habitat area. And as you go up, you have this reduction in the size of the habitat, so that by the time you get to the alpine, it's a relatively small habitat that you might have seen in the montane forests at lower elevations. So climate change is pushing those habitat zones higher and higher up the mountainside. And as a result, we're seeing a reduction in the amount of subalpine and the amount of alpine habitat that there is. The mountain pine beetle, which is a native species in North America, is also having some pretty dramatic effects on the population. And this is spurred on by climate change, which has sort of altered the reproductive cycle of the mountain pine beetle, allowing them to produce more quickly. And the outbreaks of the mountain pine beetle have been orders of magnitude greater than those that we've seen in the past. The mountain pine beetle is also a cool example of evolution, and something that we see fairly commonly with insects and fungi, where it's a disease vector for blue stain disease. So that mountain pine beetle in its oral microbiome has blue stain disease in it. And when it gets onto a whitebark pine, it inoculates it with blue stain disease. That blue stain disease then spreads throughout the tree, and it starts to break down some of that vascular tissue, so that as the beetle's moving through the tree trying to eat it up, it's already broken down partially. And altered fire regimes. So this kind of links back into climate change, but certainly into to forest fire suppression, which was a common practice in the 1900s. And that changes successional dynamics. Whitebark pine is actually adapted to low intensity fires. It can survive low intensity fires well, but a high intensity fire will wipe it out. Furthermore, the scale of these fires that we're seeing is much greater than we'd usually see. Clark's Nutcracker, those birds, they can come in and they can pretty much plant out a small forest fire burn site completely with whitebark pine. But when you have fires that are hundreds of square miles, there's just no way that the Clark's Nutcracker can get into all of those burn sites, into all that burn area, and plant those seeds. So white pine blister rust, the most destructive force, has a cascade of ecosystem effects, right? Like by taking out the whitebark pine, you're taking out this key food source for so many animals. You're reducing the genetic diversity of whitebark pine so that it can't respond as quickly as we might like to things like climate change. It's causing these successional shifts. So when you throw a wrench like a disease into a forest and you wipe out a huge population of its mature trees, you see a pretty tremendous change in the forest composition. And we're seeing other species of trees, particularly spruce and subalpine Doug fir, that are starting to take over and be more dominant in these sites that normally would have been whitebark pine dominated. And because spruce is taking over in a lot of these areas, spruce loves fire. It produces a lot of this fuel. Its seeds open up under fire. So with that effect of shifting to spruce, we're actually kind of accelerating the rate of forest fire regimes. And there's unknown ramifications. I'm certain that there are hundreds of organisms on the landscape, if not thousands, or maybe even millions, that are being affected, and we just don't know. And all of these factors are compounding, and they kind of couple, and it seems to be working synergistically in a negative fashion. I don't mean to be too doom and gloom. I promise there will be some positive light at the end of the tunnel that comes out of this. So what's the history of white pine blister rust? Well, it established in Europe in the 18th century. It had a huge impact on their stone pines. And by the early 1900s, your favorite 1900s entrepreneur, probably Gifford Pinchot, brought him in on nursery stock, and it started to spread rapidly. By the late 1950s, every major commercial pine region had infection, and blister rust is still evolving. So whitebark pine, or white pine blister rust, has this complex life cycle, which I've actually reduced here to just 2 spore stages, but it actually has 5 spore stages. But these are the important ones, or important for what we're going to talk about at least. So on the right here, we have what are called our alternate hosts. And then on the left here, we have our primary hosts. So these are our 5 needle pines. We have whitebark pine, liver pine, jack pine. And what happens is the spores that grow out on, something like whitebark pine. They land on one of the alternate hosts, like Indian paintbrush, which is Castelleja, or ribes species. So these are gooseberry, currant, things I'm sure that you've seen in the woods, and pedicularis, which is, I believe it's called elephant's foot is the common name. It lands on, one of these alternate hosts. It goes through this new spore cycle, and then it produces Basidio spores, which can land on one of these pines. A key note on Basidio spores is that in some of the early literature on white pine blister rust, they believe that these Basidio spores were more vulnerable to desiccation, to drying out, and that they couldn't travel very far. And that's something that I'm sort of indirectly testing in this study, and I'll get to that in a little bit. So what are some of the restoration strategies that we've tried, and what are we trying now? Well, originally the alternate host was targeted as the weak link. So particularly in these commercial pine regions, people tried to wipe out all of the ribes that they could find. In one study that comes to mind, they wiped out ribes for 10 years, and they only saw a 4% reduction in the prevalence of white pine blister rust. So not super effective. And on top of that, it's really hard to do. And could you imagine doing it on a whole mountain, right? In ecological terms, it's just very difficult to scale. And at that time too, they weren't even aware that there are these other species like Indian paintbrush and elephant's foot that could also be alternate hosts. Reducing the mountain pine beetle population was also an idea that people had. At least take away one of these factors that's really decimating these whitebark pine populations, like at least try and help the vigor of whitebark pine in this other way. Well, that's really difficult to do at scale too. And, you have to use insecticides to do it. So that route of managing white pine blister rust didn't really work out. And it's something that, entomologists and ecologists are still working on to this day. Crossbreeding for genetic resistance is one of the newer ways that people have approached management of white pine blister rust. So if you look at this picture here on the upper left, you'll see this cage that is around these cone producing stems of whitebark pine. And the reason they're caged is so that birds and squirrels don't come in there and they don't eat all the nuts. This tree in particular was in an area that was highly infected with white pine blister rust, but this tree was healthy. So there is some natural resistance in whitebark pine populations. And it's kind of, the idea here is to spread those resistance genes. So they're crossbreeding whitebark pines that they believe to be resistant to white pine blister rust. They've planted millions of seedlings. There's been some huge restoration projects, and seedling survival is still pretty low. Inoculation with mycorrhizal symbionts is another area of research that's being worked on by Kathy Cribb's group out of Montana State University, which is a really cool idea, right? Like, I think we all know that mycorrhizal symbionts are extremely important for pine trees. In fact, you can't grow pine trees without their mycorrhizal symbionts. And they've found some early success boosting seedling survival, but there's still work to be done there. And then most recently, what's sort of, entered the paradigm as this restoration strategy is immunomodulating needle endophytes. So needle endophytes that can either thwart white pine blister rust before it gets into the disease, or it can modulate the immune system of whitebark pine so that it's more capable of dealing with white pine blister rust when it ends up there. This is an emerging field in restoration ecology. Much of the work taking place right now is happening in agricultural systems because that's where the big bucks are. But if it starts there and it ends up in ecology and ecosystem ecology, then that's fine by me. And there is some promising early evidence in both agricultural systems and in natural ecosystems as well. So back to the whitebark pine system, right? It's restricted to the subalpine, which allows us to view the distribution and the ecology of its symbionts through a really unique lens. If you look up, virtually any animal or plant, you'll get this, you can get a distribution map of it worldwide, everywhere that it's been found. That is just not the case for fungi, but it's on a completely different scale. We have, I think 150,000 species of fungi that are currently cataloged. And my favorite species estimate for fungi is 5.3 million. So we have so much work to do. The scale and the magnitude of working on fungi is just far different from working on that of animals and plants. So how do we go about testing and looking at the biogeography and ecology of microbes that you can't even see? Well, starting with fundamental theory and ecology is a great place to start. One of my favorite theories is island biogeographic theory, coined by MacArthur and Wilson in 1967. And I'm sure you've had some exposure to Darwin's birds, right? Using these island systems to look at evolution, to look at species richness, and to look at speciation happening. So what does this theory say? Well, basically what it says is that the closer you are to the mainland, the more rich in species you will be. And the larger that island is, the more species rich you will be. If you take a look at this graph right here, you can see close to the mainland, far from the mainland, and where it intersects with an island. Close to the mainland is always going to be more species rich than far from the mainland. And here we have large islands, we have small islands. Large islands are always going to be more species rich than small islands, no matter where it intersects with the graph. So the dominant theory in microbial biogeography and microbial ecology was the Becking hypothesis. What the Becking hypothesis states is that everything is everywhere, but the environment selects. Over the last 20 years, 30 years, this has been up for debate. There's been papers that have been published that would agree with the Becking hypothesis. There's been a greater number of papers that have been published that say, hey, everything is not everywhere. And in fact, microbes are dispersal limited. So this is one of the theories that I'm testing. But we have to find the right system, and we did. We found it in whitebark pine. Here's just another example, another image showing how island biogeographic theory functions and how to think about it, right? So we have this large mainland or contiguous habitat, and then we have some large islands, and we have some smaller islands, and they're varying in distance from that mainland, from that contiguous habitat. These larger islands, as the theory predicts, will be more diverse in species than these smaller islands that are farther from the mainland. In the case of thinking about microbial symbionts, it's the host that's the habitat. So the symbiont biogeography is going to be controlled by the host. And there's a variety of suitable host systems for investigating island biogeographic theory, right? You can look at hosts that are restricted to certain elevational zones. Certainly the Sky Islands in Arizona and New Mexico, they have their own unique habitat type that, is only allowing these certain host trees to be on these, mountain island ranges. Disturbance can also create a habitat mosaic. So if you have a fire that comes in, it often doesn't have this, at least a low intensity fire will not have just a homogeneous burning. It'll create this patchwork and this mosaic on the landscape. And sometimes you'll find these like little relic islands of various trees that, survived the fire. And that's another way that you can study island biogeographic theory and the ecology of, cryptic microbes. So when I was looking at maps and trying to figure out, where I would, set up my study system, some of these mountain islands in Montana and Wyoming and Idaho started to pop out to me. And I thought, man, that would be a really cool place to test island biogeographic theory, right? We can see these mountain islands that, I have overlaid now, that vary in habitat size and they vary in distance from one another. What a cool system to study island biogeographic theory. So in this image here on the right, you can see where my sites are. I have 5 sites and they vary. This gray shading kind of represents the size of that subalpine habitat that's close by. You can see these larger areas of habitat. And then some of these sites right here, these isolated mountain ranges. We have Sacagawea Saddle, which is in the Bridger Mountains, and we have the Crazy Mountains right here. These are relatively small, and they're these isolated mountain ranges. Then we have the Apsaraka Range, which is connected via subalpine to the Beartooth Plateau, which is another place I sampled. So we have to identify some terms real quick, or describe some terms real quick, so that we can have an understanding going forward of looking at this. So the first term to define is an ecotone. So this is a transition zone between habitats. And the ecotone that we're looking at is the tree-line ecotone. So where it shifts from being subalpine, where you have dominant large trees, moving into the alpine zone, where it's only small plants and flora. And then we have different ecotypes. So these are distinct forms or morphologies that occupy a particular habitat. The main ecotype that we're looking at in this study is called Krumhold. So this is that windswept form, that shrub-like form of whitebark pine. And on this image here, you can kind of see, what's going on here. You can see this lower elevation. These are normal growth. These are large trees. They're hundreds of years old. And then we have this established Krumholds. And this is the sort of legacy tree line. This is where we have older cohorts of trees that are, hundreds of years old, but they're still in this windswept morphology. And then as we're getting up here, we can start to see this pioneering Krumholds. So these are young trees that are starting to establish higher and higher up the mountainside. So this is our pioneering Krumholds, or our tree line advancing Krumholds, and our established Krumholds, and then our lower elevation sites. The reason that we had to divide it into these distinct ecotypes is because you have to, control for some of these factors that would otherwise skew your molecular data, right? When you're looking at the various species of fungi that are associated with the whitebark pine, you have to control for some of these major factors that might change the species assemblage within whitebark pine. Back to the outline, we've covered most of the study system. Now we're going to talk about objectives, and I've laid out these objectives in terms of gradients. So it's not quite a love story. This is going to be a love slide, but ecologists love gradients. It's what allows us to study so many of these theories and questions that we have about the distributions of organisms on the landscape. So gradient is simply just a physical measure that explains the distribution of organisms. And the major gradients of this study, it's going to be ecotype or elevation. Those two are tightly linked, right? Where we have our lower elevation stands being at the lowest elevation. We have our established Krumholds. That's kind of our middling elevation. Then we have our pioneering Krumholds, which are the tippy top of elevation, the farthest that trees can establish up the mountainside. And then we have this infection severity gradient. So we sampled trees that varied in infection severity, some that were highly infected and some that were completely healthy and highly infected forests, and habitat isolation. So this idea of island biogeographic theory and how isolated that habitat is. By looking across this gradient of habitat isolation, we might be able to glean out some really interesting results about how fungal dispersal is limited or how it's not limited for these particular organisms. So objective 1 is to identify endophytic fungal species associated with these 3 major ecotypes. Our gradient here again is ecotype or elevation, and we have lower elevation established crumholds and pioneering crumholds. The question we're asking with this is, do fungal communities differ between these 3 ecotypes? Objective 2 is to characterize the endophytic communities of whitebark pine associated with varying colonization levels of white pine blister rust. So our gradient is infection severity, and the question we're asking is, are there any particular species associated with healthy trees and highly infected stands? Our third objective is to examine species diversity in the context of habitat area and distance to source population, so testing island biogeographic theory. Our gradient is habitat isolation, and our question is, does habitat isolation correlate with fungal species diversity? So the more isolated you are, does that result in a less diverse community of fungal endophytes? And do whitebark pine fungal communities sampled in this study differ from those studied in other whitebark pine studies like Aaron Moeller and Ken Aho's group and Lorinda Bullington? Do their fungal endophytes from very different, from different states, from very different regions, how do they compare? So that's at a pretty large scale. And our fourth objective is to characterize the prevalence and intensity of white pine blister rust in the crumholds ecotone. So as I mentioned before, these Basidio spores, were initially viewed as, and they still are viewed as, being vulnerable to drying out, and they can't travel very far. And the theory is that at high elevations, the crumholds ecotone, that tree line upper ecotone, it should have a lower prevalence and lower intensity of white pine blister rust, according to that theory. Our fifth objective is to compare fungal community overlap between whitebark pine and spruce. So in all these sites, we sampled spruce as well as whitebark pine. And the question we're asking with this is spruce a reservoir of fungal endophytes for whitebark pine? If whitebark pine goes extant from some regions, but then it's brought back through restoration, are they going to have the fungal symbionts that they need to persevere and to survive and do well in those regions? So back to objective 1 real quick. These are, again, 3 major ecotypes that we're looking at across this elevational gradient. So do fungal communities differ between the 3 ecotypes? For pioneering crumholds, we had to sample in a unique way, and we had to be a little creative about it. It wasn't as simple as just setting up a plot, because at these high elevations where it's, these trees are moving up, they can be spread out by great distances, and they're not, it's not like a forest or a stand at that point. So what we did was we went to the highest tree on the mountain, and we recorded what that tree was, the species of that tree. We recorded whether it had white pine blister rust, whether there were any dead trees present in this vector of the mountain that we're sampling, and whether there are any of the alternate hosts in the area. And we just went down one by one to each tree sampling until we eventually ran into the established crumholds. So the main distinction here between pioneering crumholds and established crumholds is that this established crumholds is an older cohort of trees. These trees have been here for a long time. For hundreds of years, they've had this sort of climactic range at the upper edge of the ecosystem. In terms of tree selection and looking at infection severity, so you can see a whitebark pine here that is completely dead, and you'll notice that all the bark has actually been ripped off. This is kind of an interesting thing with white pine blister rust. When it's producing these Basidio spores, it produces this sac on the bark of the tree that is apparently really sweet. I've never tasted it myself, but squirrels will get into that sack and they'll start ripping it apart and they'll rip off all the bark. So this is like one of the ways you can tell, whitebark pine has been here and a squirrel has come in and ripped it up. And I imagine they're playing a pretty large role in dispersal as well. So we selected 10 trees per ecotype. We tried to select 3 that were healthy, 2 that were low severity infection, 2 that were mid severity infection, and 3 that were high severity infection. So on the image on the right, or on the left rather, you can see a high severity infection. It has over 70% canopy kill. There were obvious signs of blister rust cankers. And on this picture on the right, we can see a low severity infection. Actually, right down on one of the lower stems, there was a little bit of blister rust coming out from this, this stem that was dying off. In terms of established krummholds and the lower elevation sites, we were able to set up plots. So these were 60-meter squared plots, and we recorded things like forest diversity. So what species are present here? In this particular picture, it's pretty much dominated by whitebark pine, except for a few larches. And then we recorded on every tree within the plot, whether it had blister rust on it or not, and how severe that infection was. We recorded diversity and abundance of plant species, presence and severity of infection for all trees. If there were any dead trees, what the species of it was, the species of that dead tree, and what the cause of it was, which is not always clear, and the presence of alternate hosts. So our molecular methods have been really challenging. Generally, I consider myself a field biologist or a field ecologist, but I have really gotten a baptism by fire in genetics by working in this fairly difficult system. Needle is essentially a chemical soup of terpenes and phenols, and it makes it very difficult to actually amplify any of the DNA that's in there. These things are PCR inhibitors, so they inhibit this polymerase chain reaction that we use to get sequences out of needles or even out of a fruiting body. So we took a hierarchical approach to testing what might work, and we started with the simplest stuff, right? So needle preparation can vary, and this slide is just sort of a depiction of how many different ways you can do this and how many different variables you can tinker with. So in terms of needle preparation, you have to sterilize it, right? You don't want any of the sequences from the epiphytes to be on there, or spores that have landed on there. You have to grind it in liquid nitrogen. You can bead it. There's essentially, each one of these represents its own variable that you can tweak when you're trying to get the fungal genes out of a pine needle. And then DNA extraction, we tried a variety of methods. We tried a basic extraction, so sodium hydroxide. We tried various CTAB protocols. We tried gene clean, which is a type of kit. We tried rapid one-step extraction or rose extraction. And we tried these QIAGEN kits too. And what's not listed here is the kit that actually worked the best, which comes from Nucleospin. It's the Nucleospin Plant Power Pro 2 kit, I believe. So we have 5 variables that we can tinker with here. And then we can purify the DNA as well. We can use these phenol chloroform steps to try and purify it. We can purify it once or twice or 3 times. We can do an ethanol precipitation to try and concentrate that DNA. And then there's all of these different things that we can tinker with when it comes to the actual PCR. So how many are approaches? Well, just like super quick and dirty math, there's 360 approaches, but that doesn't even begin to to describe how many there really are when you really consider all the variables you can tinker with. So we tinkered and we tinkered and we kept tinkering. And eventually we started to get, these positive bands. So if I can, or these positive results. So if I can draw you to this picture on the left here, what you'll see is this faint row of bands on the bottom. These are primer dimers. So what a primer is it's a piece of synthetically constructed DNA that is going to attach to a particular part of the fungal genome that you want to amplify. And what can happen is these primers can come together and bind to each other. And these are called primer dimers. So that's what you see in this lower band. And then in these upper bands, this is where we're actually seeing positive amplification. And there's sort of a third thing I don't want to bring up, which are artifacts. But you can ask me about that if you'd like. So we started to get some positive results. And, this is just a glimpse into so many of the different tests that we ran, different PCR protocols and different extraction methods that we tried. Now we'll take a look at what we found in our initial field results, looking at this whitepine blister rust prevalence in the subalpine. I can see I'm already going long. Nobody has ever let me talk for this long, so I'm kind of in new territory. So I might move through this a little bit quickly.
Don't worry, Dylan, you're all good.
I'm all good? Okay, all right. All right, we'll take our time. So how does disease prevalence at high elevation stands compare to low elevation stands and to what has been found in other studies? Well, all right, here's this complicated table. Let me key you in on these boxes real quick. So these are highlighting our pioneering crumholds. So this is our highest elevation stand. This is where we would expect, based on the theory of Basidio spores being more vulnerable, to have a lower percentage of whitebark pines that are infected. If you kind of just look across, you'll see that 36% are infected, 0% infected in this pioneering crumhold stand, and 17% and 30% here. So how does this relate to the previous literature and what we would expect to see? Well, it is a little bit lower, just like eyeballing it, but the statistics that we ran on it suggest that there is no difference. And that kind of makes sense, right? Like if you look at, what we're seeing here, we've got another 0 that's in our lower elevation stand. We've got 48%, then 35%. So it wasn't a clear enough pattern for the statistics to elucidate any information. So ultimately, it's in line with other studies. And if you want to get more information on that, you can see a Ressler and Tombach paper from 2008. There's one other really important piece that we can get from this really, complicated table if you stare at it for too long. So let's simplify it. We can see what stands are going to be the best for comparing our endophytic fungal communities. So what are we looking at here, right? If we look at tree density across these established crumhold sites, we see that they're relatively similar. This first one's a bit of an oddball, but otherwise relatively similar. We see that community composition sort of has this mixed dominance between this PA stands for Pinus albicollis, that's whitebark pine, our subalpine fir, that's AL, Aves lasiocarpa, and then our spruce, which is Pinus engelmannii, I believe, Engelmann spruce, right? So we see that there's a relatively similar community composition, and we see that there's a relatively similar percentage of whitebark pine with blister rust symptoms. So I think these established crumhold sites are going to be really the key to elucidating any information on island biogeographic theory and some of these patterns that we see in species diversity. A little bit more on molecular work. One of the first things you need to do when you get your molecular work is to make sure you saturated something called a species accumulation curve. So if I can draw your attention to this graph, and specifically to these lower number of samples. So if we take a look at 5 samples, you will see that these samples or these sites didn't plateau out quite yet, right? This one is still rising. But if you go to the larger number of samples that were taken, you can see that plateaus are beginning to form, and that means you saturated your species accumulation curves. So hopefully we saturate our curves. I think that we will. Running the math on it, we have 30 samples per site. That's with some nesting. Sorry to bring any statistical PTSD up. And we have 10 samples per ecotype per site. We're using Illumina MySeq, which will give us 15 million reads. And we're looking at 3 loci. So loci are this particular portion of the genome that we want to amplify. And we have about 300 samples. We have about 150 whitebark pine samples and about 150 spruce samples. And that puts us at 48,000 reads per sample, or 16,000 reads per sample per loci. Generally, what you're shooting for is 10,000 reads per sample. So we should saturate our species accumulation curves, just doing the quick math on it. In terms of biogeography and distance, testing this island biogeographic theory, you can see this figure right here. This is from Helander and colleagues in 2007, where they were looking at endophytes in the Finnish islands. And I believe they looked at 17 different islands that varied in isolation and varied in size. And what they found was that the farther the distance from the mainland, the lower the species richness. So this is on the y-axis, we have a log scale going on. So the farther you get from the mainland, the lower the species richness. And this comes back, to island biogeographic theory. It agrees with it in these results, and there are certainly a number of studies that agree with it as well. To habitat size, does that correlate with species richness? Well, right, like we would think the greater the habitat size, the greater the niche diversity. So we should have more organisms on larger habitats. In statistics and ecology, we use these things called a species area relationship, or a SAR. And Kabir Pay's lab at, I guess he was at Berkeley at the time, with Matteo Garballato and Tom Bruns. They ran this cool study in Bishop Pine, where they were looking at island biogeographic theory in mycorrhizal species. And we have another study that was done using island biogeographic theory. That's this Helander study, where they looked at fungal endophytes in birch through the lens of island biogeographic theory. And all right, what did they find? Well, they found that as land area increased, both of them have these upward trends. As land area increases, so does the log of species. So what about this biocontrol potential, this ability of fungal endophytes to modulate the immune system or to thwart disease? Well, Bullington and colleagues laid the groundwork for some species of interest. We're also going to run what's called an indicator species analysis. So it's basically, this way of highlighting, are there any species in these assemblages that we get back that might be worth taking a closer look at. They found these 2 species in particular that were negatively correlated with overall disease severity. So those are 2 that we're going to key in on. And then Metarhizium anicifolii, which was correlated with a lower number of needle spots and blister rust cankers. In Western whitepine, Ganley and colleagues, they inoculated these trees with endophyte slurries. Then they inoculated trees with whitepine blister rust. And well, they found that trees that were inoculated with endophyte slurries, they lived longer. They had a reduction in blister rust severity. So it seems like there is good biocontrol potential. I should probably define what a biocontrol is. So a Biocontrol, as the name suggests, it's a biological control agent. So it's something that we can enter into an ecosystem or an agricultural system that doesn't require insecticides or fertilizers or anything like that. You know, it's natural in that sense. It's of the biological nature, but we can use it to control disease or to control, you know, things like drought. So our overview and outlook, whitebark pine populations are at extreme risk. It is a sad state that we're in because the whitebark pine and so many of these 5-needle pines like the bristlecone pine are iconic trees. The habitat islands of whitebark pine provide this really unique system to look at island biogeographic theory. And fungal endophyte communities may be structured by biogeography. And we may find that, you know, it contradicts the Becking hypothesis that everything is everywhere. But I think the literature is really moving towards the idea that fungi are dispersal limited, but to what degree and at what scale are they dispersal limited? And is it biologically relevant? And it might be relevant for some species that are relatively homogeneous on the landscape, but or I should say it may be irrelevant for species that are homogeneous on the landscape, but it may be very relevant for species like whitebark pine that have this patchy distribution. And these resistant trees may harbor mutualistic fungi. There may be a very legitimate restoration strategy in using fungal endophytes, and I think it's a super exciting aspect of the field right now. So, right, it might be the cryptic link for whitebark pine preservation. I would like to thank my advisors, Tom Volk, Todd Osmundson, my mentors, Meredith Thompson and Kathy Cripps, my lab and field assistants, Liz Dominguez, Holly Critter, Chance Knopfsinger, and my funding sources, the University of Wisconsin-La Crosse, the Oregon Mycological Society. Thank you so much. I appreciate it so much. And the Sonoma County Mycological Association as well. These are my references. I'm happy to send these out to anyone that's interested, or if anyone has any further questions, like that we don't get to during this follow-up discussion, shoot me an email. I'm so happy to talk about this stuff. So I think we're on to the questions. I'm only 13 minutes past time. I think that's not terrible.
Thanks, Dylan. That's a lot to digest. I've been many years out of my own work, and a lot of those ring a bell coming back to it. So it sounds like, have you done 1 year's field sampling, and do you plan to do another year's field sampling, or is it just a one-time sampling event?
It was as much as I tried. It was just a one-time event. You know, I was cooking up new ideas to get back out there, but yeah, it was one-time event. I think we got the data that we needed. There's always more that can be done, and it's obviously a beautiful landscape too. So, you know, love to get in the field and travel. But yeah, 1 year.
Okay, and yeah, that is a great area. I can't imagine how wonderful. You took advantage of the time you were there, right?
Yeah,
yeah,
of course.
Definitely. So I guess, one of the questions that came up for me, and I think you touched on it at the end, you said, because a lot of your results aren't in yet, I guess I'm just kind of asking some of your gut feelings. Did you have a number of stands or plots that you had where you saw a number of trees that had low disease prevalence, but were in plots that had high disease prevalence?
Yeah, it can be really sporadic. I mean, I did, when I went into these ranges, I spent 2 weeks just driving around to potential sites and going up into them and looking at what the disease severity was. It can vary dramatically. You can go into some areas and it's like every tree is infected, and there doesn't seem to be a lot going on, like a lot of new seedlings establishing. And then other areas you go into and it doesn't seem like there's that much blister rust going on. You know, particularly I went down to Wyoming and Colorado, and it didn't seem to be as infected as Montana, which I thought was really interesting. I tried to select sites that were sort of in the middle, that weren't like highly infected, you know, where it was like 90% die off. And part of that is just because when you're, you know, setting up your plot and your sampling method, like if you have to walk 2 miles to find 2 or 3 trees that are healthy, like completely healthy, that makes for a really long day. So I tried to pick sites that, you know, sort of had this middling range of infection severity, or what I felt like. So yeah, that was kind of my approach.
Okay. And so the key that I'm hearing is you've taken the samples of just the needles, or are you looking at bark as well, a cambium layer or anything like that?
I'm looking at just the bark, mostly because of the mechanism of white pine blister rust infection. You know, I don't know if I said this, but what happens is it lands, the spore of white pine blister rust, it lands on a needle, and then it grows a germ tube through the needle stomata, and then it enters into the vascular system and it spreads. So that needle seems like it would really be the first line of defense, and whatever living in that needle would be the first line of defense. It rarely enters through the bark. There has to be sort of some strange circumstance, like either a beetle, you know, sort of opens up that and then a spore lands on there. And there really isn't much in the literature to go off of that. But bark endophytes are a totally understudied area that I hope some students and some other researchers start studying, especially when we talk about things like the mountain pine beetle, right? So yeah, I just took needles.
Okay, we have a question from Ayer. She's asking, do bristlecone pines have the same problem?
Not that I have come across. I have really been made more aware of limber pine, lodgepole pine, and whitebark pine being the primary species that are infected by this, with limber pine and lodgepole pine. And lodgepole is, it's not even a 5-needle pine, it's a 2-needle pine, but it has been found on it. And yeah, so it varies in the 5-needle pines how badly it's infected. I haven't heard about bristlecone pine being one that's, you know, being attacked by it, but I know there's some other issues going on with bristlecone pine, particularly forest fires. So, okay.
And thank you for clarifying on how it gets into the tree. My experience is on the East Coast with white pines. You know, we had a number of them on our property, and we were trying to manage for it, and so we never knew how it actually entered into the pine. But I just remember seeing those big pustules, I guess, for lack of a better word, of that orange rust that was out there. And you're right, the porcupines loved to eat around that as well. That was a big problem.
Yeah, I've been wanting to like try to make a syrup or a tincture out of it or something like that. I'm not sure if it's toxic, but if the squirrels like it, it can't be that bad. I've heard it's very sweet, so they must be concentrating sap or something like that.
So as you're moving on, are you taking samples of the spruce as well to see what sort of fungal endophytes are in there? Because you did touch on that, thinking that the fungal endophytes in the spruce or the surrounding habitat might be a key.
Yeah. So in all of these sites, we took, we ended up with like 150 spruce needle samples, and yeah, from all of these same plots. So if whitebark pine, is diminished to a great degree in some of these areas, maybe spruce can act as a reservoir of fungi for whitebark pine when it starts to make its comeback. And I think it touches on some interesting stuff in fungal conservation, because fungal conservation is a really difficult thing to think about when you can't see the organisms. So yeah, hopefully, I guess I do hope that there is a really large amount of overlap between spruce and whitebark pine, even though that makes testing certain theories really difficult. So yeah, we're looking at spruce and we'll see what we find.
So it sounds like a lot of what you're doing is really based on theories in the literature and trying to find out, to test them in this scenario. I guess I'm seeing the applications more agricultural, but commercial. You're talking about natural regeneration of the whitebark pine. Are you ever foreseeing something where they're actually, like you were talking about, is hybridizing them and then replanting them?
Yeah, so I actually had the chance to go out with Kathy Cripps and some of her students, Chance Knopfsinger, and we went to these, she was working with these restoration groups where they planted whitebark pines that were inoculated with mycorrhiza. And these restoration projects can be huge, like millions and millions of trees. And they did a subset, I can't remember how many thousands of trees that they planted that were mycorrhizally inoculated. But there might even be, we might find that certain regions have endophytes that, modulate the immune system, thwart the disease, but they're not present in these other ecosystems. So then it becomes a question of how can we get them in there so that they can kind of do their own thing and spread. I don't necessarily envision like a crop duster coming and like spraying an entire forest, but I think there might be a seeding strategy that can be applied where, we kind of just get it in there and we let it do its thing. And a lot of this seems to come down to buying the whitebark pine time to evolve. So, these trees, they don't reach, seeding maturity until they're like 50 years old or 100 years old. So if we kill off, or, if blister rust kills off, a huge percentage of the mature trees, it doesn't leave a very big bank for naturally regenerating whitebark pines to keep establishing. So I think if we can buy it time and give it time to evolve, that would be a really good step in the right direction.
I guess that kind of leads into the next question I had was, are you seeing the disease across all ages, including young saplings or seedlings?
It seems like I would say yes. In Lorinda Bullington's study, they used, relatively young trees, young whitebark pines, and they inoculated them with white pine blister rust, and they got white pine blister rust. In general, from talking to people and from what I've seen in the field, is that it seems like younger trees, like saplings in particular, are less likely to get it. But that's anecdotal. I haven't done that myself. I would have liked to have found out the age of all the trees that I sampled. But we're talking about like taking a cookie, from a threatened tree. So there's some really cool ways to do it where they're like combining sort of like allometry, which is like the partitioning of growth, where they just take like a cookie from the stem and then they run it, like they enter whatever that number they come up, like the stem they find is 10 years old, and then they kind of run it through this allometric equation, and they're able to figure out how old the tree actually is, which is really cool stuff. It's not something that I did. So yeah, your original question, yeah, I guess all I have is like my anecdotal evidence on it. So.
That totally makes sense. And then how long, I guess it depends on the severity of the infection, but how long would it take to kill a tree? I guess that depends on, you were saying the squirrels eating the cambium layer off, mm-hmm. if that happens.
Yeah, so this is, kind of something that's definitely entered the restoration realm, because generally it takes 3 to 7 years to kill off the tree. Like a lot of times what you'll see happens is the blister rust gets onto the tree and then it kills off like a major part of the tree. Like it'll kill off like a whole branch, right? And then it'll get re-inoculated and it'll kill off like another 2 branches. And in the meantime, like trees have this response to disturbance where when they're under, severe stress, they seed out. They just produce as many seeds as they can because they know that the end is near. So one of the ideas here is like in this restoration realm is trying to like improve the vigor of the tree so that if it normally would have died in 5 years, maybe it can last for another 10 years where it's still producing seeds. And I don't understand all the fine details of that, whether it's like cutting trees off or going in there and doing some selective cutting to remove trees that are infected, but that's something that people are certainly thinking about.
Okay, we have another question. Is the current thinking that what was once believed to be genetic resistance really the presence of the protective fungi / endophyte species?
Yeah, that's a really interesting question. I would say that the predominant thinking is that it is genetic resistance from the tree. However, there's things that are commingling, because the tree, the genetics of the tree has a huge role on what that endophyte community is, because you might get 2 trees that are right next to each other, but they came from different parents. And one of them is producing 34 different kinds of terpenes, and the other one is producing 20 different kinds of terpenes. And those terpenes are really the sort of molecular gatekeepers of what endophytes are going to be surviving in there. So it might just be that the genetics of the tree are altering what endophyte communities they have. It could be that. There's this paper about a novel canker or novel aspen canker in Alaska. The names of the researchers escape me right now. Is at Bonanza Creek LTER, and they looked at the genetics of the aspen canker, or of aspens that were being infected. And it's like 80% of the aspens in Alaska are being infected, and it's one of 5 deciduous trees up there, so it's like a pretty big deal. And they don't know why. They don't know why 20% of these trees are surviving. So there are some suggestions that maybe it's endophytes, which is really interesting.
There you go. You're going to have someone else looking at that as well. Another question, Joe. Can you compare the pathogens causing whitepine blister rust and those causing spruce needle cast disease?
can I personally compare them and how they're
or maybe discuss the difference?
Yeah, I can't because I don't know enough about spruce needlecast. I can't. I just have to bow out on that one.
That's okay. That's okay. So tell us about your next 6 months. What is it with your research? What's going on? Did you finally receive the machine you needed?
So it was really reagents and kits. Kaijin had back ordered me like 6 months on the kit that I really wanted to use, and then I ended up getting some test kits from Nucleospin and tested them out, and they worked, and they sent me a bunch more, and they've been working great. So really over the next week, I'm going to finish up preparation for our Illumina libraries, and we'll send those in for sequencing, and in a couple, 2 or 3 weeks, I'll have those back, and then we can analyze the data and figure out what these endophytes are and what they're doing.
Good. Do you have, you talked about other people doing similar research. Do you have someone in your lab or, close to you that's doing something similar with other species?
Yeah, actually, one of my really close friends is looking at endophytes in hemp, and he's looking at wild hemp versus cultivated hemp, and the sort of applying a similar infection severity gradient with some of the hemp molds. And basically they're trying to, he's working with a few other groups that have financial interest in, producing, hemp. And they're interested, like, can they spray an endophyte on there? Because, spraying insecticides and fungicides on hemp is not something that they want to do. So they're looking to see if they can find endophytes in them.
All right. So obviously, these sorts of things, when you work together, there might be a lot of cross thought going on.
Yeah, there's like a ton of awesome conversations that we have, and that's Caleb Casper that's working on that stuff, and then my advisor Todd Osmundson, he did some work in I think it's a Polynesian island in Moorea, where he looked at like, basically, I think they sampled most of the flora on this island, and not so much on the infection, pathogen disease side of things, but on this island biogeographic theory. And he's really interested in, biogeographic controls of fungi. So, there's like a lot of crosstalk going on, and it's fun, and, it just enhances, your work. So
then once you get this done and write the analysis, when are you thinking of defending your thesis? Do you know yet?
Yeah, it's looking like mid-January. So yeah, there will still be a lot of writing left to do after that. But my advisor, my committee members are kind of like, just get the hell out of here, man. Finish your master's, and you can work on, really buttoning up the manuscript after that. So yeah, hopefully defend in January.
And then what? What's now that you've had a taste of this, what piques your interest?
I've been so steeped in endophytes for these last couple years that even when I try to think about other ideas, I keep getting drawn back into endophytes. And I think there's so much green pasture, for a researcher to tackle. And I think one of the fields that hasn't really been touched yet within endophytes, and endophytes are understudied and blossoming, is how they affect, these entire ecosystems, right? Like, really the ecosystem ecology of it, just thinking about, whitebark pine, right? And these massive population losses, like, that's a lot of carbon that's being taken out of the system. needle endophytes are, the vast majority of them are latent saprobes. So they kind of exist in the needle and they don't really do anything. They don't really help the plant. They don't hurt the plant. The plant just lets them be there. That's most endophytes. And they're just waiting around for that needle to die. And then they're the first one that can decompose it. So they're also playing this huge role in duff and litter accumulation, which obviously has to do with fire regimes and all of that decomposition and carbon turnover and nutrient turnover. So I think that the sort of coming together of ecosystem ecology and endophytes is like ripe for the taking.
Well, maybe we'll have you back once you get through this and can tell us the results of what you find. As the last parting is, from a kind of a neophyte, someone who doesn't know much about this, how many could be in one tree or in a needle? You talked about 7 at the beginning, but
yeah. So it was part of George Newcomb's group. I can't remember what study it was, but they found, I believe, on average, there were 7 fungal species per needle. And they were finding like 7 to 14 was sort of that, average range of how many species are in a needle. I'd have to touch up on that paper, but there can be a lot in a needle and there can be thousands of different species inside of a single plant, so.
Okay, well, with that, we especially want to thank Dylan for staying up late. You're what, 2 hours or 3 hours difference from us?
2 hours, but
2 hours.
Think about the time change.
I know, I'm
like
so sorry.
Hours.
So sorry. we did that to you.
It was bad timing.
Yeah, but we wanted to thank you for taking the time and wish you well on your research. We're so glad that we could support you. And to everybody, this is our last presentation meeting for the year. We hope to have some sort of survivor banquet, possibly virtual, however we could do that in these strange times. And then next year we start again in February with a scholarship speaker. And then in March, we will have Trad Cotter, who will talk to us about some of the cool stuff that he's actually doing. So with that, I just want to say thank you and good night.
It was great to speak to you all. I wish I could have met you in person.
There you go. Let's see.